In this video, we're going to focus on skeletal myofiber anatomy, or muscle cell anatomy. Recall in the previous video that we went over the whole muscle structure and the organization of the muscle. Within the muscle, remember you have bundles of muscle cells, and these bundles are called fascicles. And this is a muscle cell or myofiber here within the fascicle. If we magnify this muscle cell, it looks like this right here. And what we're going to see is that a muscle cell consists of a muscle cell membrane, which is known as the sarcolemma. In muscle, we're going to see a lot of the terms begin with sarco. Sarco is a Greek term that means flesh. In fact, when we're talking about, remember Egyptians, what they put their bodies in, their dead bodies in? Those are called sarcophagus, sarco meaning flesh. Within the muscle cell, we have the cytoplasm, but we call that the sarcoplasm. And we have lots of what are known as myofibrils. Now you got to be careful on this because a single muscle cell is called a myofiber. But within the muscle cell are myofibrils. And myofibrils are the contractile proteins for the muscle cell. And these myofibrils consist of myofilaments, thick and thin myofilaments, which you see here. Here is a scanning electron micrograph of a muscle cell. And take a look at this. This is the sarcolemma here, the muscle cell membrane. And within this muscle cell, you see lots and lots of myofibrils. Let's get into the details now of myofiber anatomy. As I said, we have the muscle cell membrane. Muscle cell membrane is called the sarcolemma. Within the muscle cell, you have lots of cytoplasm, which we refer to as sarcoplasm. Sarcoplasm has mitochondria within it. For energy, of course, you're going to use lots of ATP, so you need to have lots of mitochondria to provide that ATP for muscle contraction. There's going to be lots of glycogen granules in here, which glycogen is a storage form of glucose, and we use that glucose as an energy source. There are lots of myofibrils within the muscle. This right here is a myofibril. Here's another myofibril. We're going to see that the myofibrils consist of two main types of filaments. We have a thick filament, which looks like this, a little thicker than the thin filament here. And it's the interaction of these thick and thin filaments that are going to allow a muscle to contract. We're going to see that the thin filaments slide over the thick filaments during a contraction. Well, these thin filaments here are attached to the Z disc. I call it a Z disc because it looks like, well, a Z, little Z lines. And from one Z disc to the next Z disc is a functional unit of contraction in skeletal muscle called a sarcomere. Throughout the muscle, we're going to see that there are these T tubules, and T stands for transverse because they're going in a transverse direction within the muscle cell. And these are tubular invaginations of this sarcolemma that extend through the cell. These T tubules contain the same components as the sarcolemma here. They're going to have voltage-gated sodium and potassium channels. So when that action potential comes down that sarcolemma, it's going to continue down through the T tubules. And this is really important because we're going to see that action potential as it travels down the T tubules is going to stimulate this organelle called the sarcoplasmic reticulum to release calcium. And calcium is absolutely essential for muscle contraction. And the T tubules is how we get the action potential deep inside the muscle cell. Within that muscle cell, you're going to find lots of nuclei. Remember, skeletal muscle is multinucleated. Skeletal muscle cell has lots of endoplasmic reticulum. And this endoplasmic reticulum is in and wrapped around the myofibrils. Here's a myofibril. Remember, a myofibril has thick and thin myofilaments. The sarcoplasmic reticulum has these structures at the ends, which are right up against the T tubule. So here's a sarcoplasmic reticulum, and it has a dilated end sac here and a dilated end sac here. And we call these dilated end sacs, we call these terminal cisterns or terminal cisternae. And they lie on each side of the T tubule or transverse tubule. The interaction of that action potential that comes down the T tubule is going to stimulate these terminal cisterns to release calcium. We call this T tubule, flanked on each side by a terminal cistern, a triad, because it consists of three structures, the T tubule and two terminal cisterns. You can refer to these as a terminal cisternae, or you could just call them terminal cisterns. The arrangement of that sarcoplasmic reticulum, you can see in here is a thick filament and a thin filament. And remember, the thick filaments we're going to see are going to pull the thin filaments over the top of them. They're going to slide them over the top of them. And that's going to allow for a contraction. There's going to be a lot of interesting physiology that's going to come once we know about these structures. Let's go ahead and pause the video and test your knowledge by labeling these structures. Here are the answers. Pause the video and label these structures. And here are the answers. Let's test our knowledge. Go ahead and pause the video and select the term that makes the sentence correct. Here are the answers. Pause the video, select the term that makes the sentence correct. Here are the answers. So now we're going to go into more detail about these thick and thin myofilaments that make up the myofibrils. And we're going to see that the interaction of these thick myofilaments here with the thin myofilaments here are going to allow these thin myofilaments to slide over the thick myofilaments. The end result is that you're going to have contraction. You're going to have the sarcomere, which goes from the Z disc to Z disc, gets smaller. So we're going to see that the Z disc here to Z disc, this line represents a Z disc, to this line represents a Z disc. These sarcomeres are going to shorten because the thick filaments are going to pull the thin filaments over the top of them. And as they pull it towards the center, this thin filaments get pulled towards the center, and this thin filament gets pulled towards the center. That's going to cause a contraction. So we look at the myofibrils. Well, these are rod-like structures that extend the length of the cell. So we're looking at this muscle cell here. These myofibrils go all the way down that muscle cell. Myofibrils are composed of cylindrical bundles of thick and thin myofilaments arranged in repeating units called sarcomeres. And that's just right here. This is a sarcomere. Here are our thin myofilaments. Here are the thick myofilaments. And these are arranged in cylindrical units all the way down the muscle cell in sarcomeres. Here's one sarcomere. Here's the next sarcomere. Remember, the ends of a sarcomere are the Z discs, to which the thin filaments are attached to. And it's the interaction of these proteins that allow for muscle contraction. We're going to focus now on the thick myofilament. So the thick myofilaments are composed of the protein myosin. And basically, a single myosin protein looks like two golf clubs whose shafts have been twisted about one another. Here are the heads of the two golf clubs. These are the shafts of the two golf clubs. See how they're twisted about one another? Well, you take about 300 of these myosin molecules. These are joined together to form a single thick myofilament. That's what it looks like here. And these heads are coming out all around that thick filament. Now, when you look at something like this, a two-dimensional structure, it shows a thick filament with these myosin heads, also called cross bridges, that go across and interact with the thin filament. But it's only showing one set of heads here. Remember, these heads are all around, not just on the top and on the bottom. Now, the myosin heads, or also called cross bridges, are going to extend toward the thin filament. And to show a more three-dimensional arrangement of these thick and thin myofilaments, here's a scanning electron micrograph that's showing a myofibril. Remember, myofibrils consist of those thick and thin myofilaments. So we have a thick myofilament, and around that thick myofilament are actually 6 thin myofilaments. It's only showing 1, 2 on this side and 1, 2 on this side, but this is just a two-dimensional structure. Three-dimensionally, you're actually going to have 6 of these thin filaments around the one thick filament. And since we have myosin heads all around that thick filament, that's going to allow those myosin heads to form cross bridges all along here to interact with those 6 thin myofilaments. And looking at the scanning electron micrograph, take a look at this. Here is one thick filament, and what do we see around that thick filament? We've got 1, 2, 3, 4, 5, 6. They're going to interact with that thick filament. And then this one over here, this thick filament, also shares the interaction of the thin filaments on this side. So they're all sharing with each other. So there's a really neat arrangement of these thick and thin myofilaments. Each myosin head contains an ATP binding site. and an actin binding site. So we look at the myosin head, we have an ATP binding site, and we have an actin binding site. The protein in the thin filament that the myosin head is going to bind to is actin. That's these purple grape-looking structures that are coming through here. So we're going to see the myosin head is going to bind to it, and it's going to pull it. It's going to slide it over. Well, to do this, we need energy, and that energy is going to come from ATP, or adenosine triphosphate. The ATP binding site, now the ATP binding site we find on the myosin head functions as a myosin ATPase enzyme. So when that ATP binds to it, this enzyme is going to split ATP to ADP and phosphate. And then when you do that, you release energy, and that energy is going to be used to cock the myosin head back so that it can then bind and power stroke and pull the thin filament over the thick filament. Now, it's hard to see that with this diagram here, so I'm going to show you an animation where we see a myosin head here interacting with the active site on the thin filament. Well, it's going to bind and power stroke and pull the thin filament over the thick filament. Let's take a look at this. So you see how it's binding and power stroking, pulling the thin filament over the thick filament. Well, what do you use to energize that head? Well, that's this green dot coming in, and it binds to the myosin head. That's ATP. And when ATP gets broken down to ADP and phosphate, which you can see the small piece coming off, that's the phosphate, and then ADP comes off. There's a whole lot of neat physiology going on here. Before I can talk about that, I need to go over the structures of the thin filament. That's what this myosin head is binding onto, is the thin filament. So we're going to next go over the structures of the thin filament, and then we're going to put it all together and talk about the physiology of muscle contraction. Thin myofilaments. Well, these are composed of 3 different types of proteins. We have actin, which is a main component, and that's what you see here in these purple grape-like structures. We have troponin, which are the red structures, and we have tropomyosin, which is this blue strand here, tropomyosin. Actin myofilaments. We call this F-actin, and it's a polymer formed of 300 to 400 globular subunits, which are called the G-actin. When you look at them individually, they're G-actin. You put them all together, we have F-actin, and these are arranged as a double row of proteins twisted to form a helix. On each of these actin subunits, there's a myosin binding site, so that myosin head can bind to the actin. Under resting conditions, the myosin binding is prevented by the troponin-tropomyosin complex. So we have here, which are shown in gold, troponin. We have tropomyosin here that's attached to, and basically the troponin-tropomyosin complex covers up the active sites so that the myosin head can't bind to the actin. Under resting conditions, this myosin binding site is prevented from binding to the myosin head by the troponin-tropomyosin complex, which covers over that active site. And if you remember from that animation I showed you a little earlier, there's an ion that's going to bind to this troponin to move that troponin-tropomyosin complex out of the way so that the myosin head can interact. And that ion is called calcium. When you look at the tropomyosin, what this is long strands of protein molecules that are located in the grooves of that actin helix. And this covers the myosin binding sites and prevents actin-myosin interaction. Well, troponin does cover a few of the binding sites, but it doesn't do it as well as tropomyosin. Tropomyosin is a main covering for these binding sites. When you look at troponin, this is going to bind to tropomyosin. And troponin bound to tropomyosin forms a complex known as the troponin-tropomyosin complex. Troponin is a complex of 3 proteins. We have troponin I, which is shown here. Now, troponin I, it's I for inhibits, because this can inhibit cross-bridge binding to actin. But it doesn't do it as well as tropomyosin, but it can do a little interference from the myosin head binding to the actin. So we call that troponin I. Troponin T, which is here, is the part of the troponin molecule that binds to tropomyosin. So since it binds to tropomyosin, we call it troponin T. Troponin C is the part that actually binds to calcium ions. And when it binds to calcium ions, it's going to cause a change in shape of this troponin-tropomyosin complex to move it out of the way so the myosin head can bind and power stroke. We call it troponin C because it binds to calcium. When your muscles aren't contracting, when they're at rest, the intracellular calcium ions is really low in the sarcoplasm. It's because all that calcium is being stored in what organelle? Sarcoplasmic reticulum. So let's test our knowledge on thick and thin myofilaments. Go ahead and pause the video, select the term that makes the sentence correct. Here are the answers. Here's part 2. Go ahead and pause the video, select the term that makes the sentence correct. Here are the answers. Part 3. Pause the video, select the term that makes the sentence correct. Here are the answers. That concludes this video, and let me know if you have any questions.